Hello friends, good evening. I'm Pratap Bhosle. We are back with our 19th lecture in the series. That is an transcription today. So in the next 4 to 5 lectures, we will just complete the entire cell biology. Then we are starting with this physiology module, the next chapter in this physiology around 1st of December. Okay. So let's wait one more minute. We will start exactly at 5:03. Till then, I will just introduce to some of the other courses are there, right? So you can join this Telegram group if you are watching this first time on the YouTube, or you can just log into the website. There you will find the free courses are also there and paid courses are also there. So you can join those free courses and you will get all of these lectures till 19th in that only one course page only. So I have already started my cell biology module on the 6th of November, and it is about to get complete around 22 of November. So if you want to just have an even the recorded lecture of the cell biology along with the printed notes, you can even join it now also, or you just wait for my next physiology module, which is going to be start around 1st of December. Okay. So yes, we will start with our today's lecture, that is about this transcription. Yesterday we have seen this DNA replication. DNA replication is. Tomorrow our lecture is would be. Yes, good evening, Shravani and Arya Jadhav. Good evening, all of you. Let's see the next thing. RNA is synthesized. See, the transcription is what we have seen. This DNA replication. Okay, we have seen this DNA replication. What is happening in this DNA replication is this DNA. What was earlier was there, its copy is being made while this DNA replication. Okay. Now what we are seeing is that how this copy of DNA can be transcribed into the mRNA. So the transfer of this DNA into the mRNA is called as what? Transcription. Okay. Tomorrow we will see about the translation. Now what is meant by translation? Translation means nothing but this mRNA will be translated into a protein, any particular protein. Okay, so that is the case with the translation. Now let's start with this our first type of or first category of this transcription. Shravani is saying, from where should I start, sir? This is the first time I am watching the video. Please help me. See, Shravani, if you have such query, you can ask these queries at the end of this session. Okay, because we cannot break this link of the entire lecture. I will answer your all of your query. Just ask it after suppose 20 minutes because it will require at least 20 to 25 minutes to complete this lecture. So let's start. See, this initiation is the first step, and you will find this is the prokaryotic transcription is there. This is this one. Okay. So in this prokaryotic translation, we are going to see the first, the initiation phase, then it will be the elongation phase, then it will be the termination phase, and I will explain all those three phases or all those three steps with the help of this diagram also. Okay. So let's first understand that transcription in the terms of theory. Okay, so this is the initiation. What is the requisite is there to for start this initiation of the prokaryote transcription is first the sigma factor. Obviously, the first is RNA polymerase. The second would be the sigma factor, and there are certain promoter sequences are there, right? What are these promoter sequences? Suppose this is that the region of promoter sequences here. Okay, here where this entire proteins will get attached, right? Here this is the DNA strand, right? And on this region only there is a region which is called as promoter region. And what is this promoter region will have? This promoter region will have this kind of promoter sequences like Primbo box, also called as Tata box, and the dash 35 minus 35 sequence. Okay. And what are these showing? This Primbo box can also be called as this minus 10 sequence or dash sequence, 10 sequence. Why it is called as 10 and why it is called as 35? The reason is their location. Okay, the location of that Primbo box and location of that 35 sequence is minus 35 sequence is at the distance of 35 nucleotides from the initiation. Suppose this is the initiation point. Okay, this is the point where the transcription is going to the start. From that point, this location of this rainbow box is 10 nucleotides, and from this point, the location of this the sequence it is 35 nucleotides away. So that's why they are named as -10 and -35. Okay, so these will be the initiation factors. Okay, the first one is what? RNA polymerase. The second is what? Sigma factor. Okay, you will just have to go through this theory first, then I will just show you the exact visual diagrams or exam diagrams of all of these. The next will be the sigma factor. Third one is this, and third and fourth one are what? This Prinbo box and 35 sequence. Okay. Once we have this, we are able to initiate this our entire protein synthesis. So now let's this we have just initiated. Now we are just initiate the whatever these amino acids we have or whatever this mRNA we have. Just initiate that strand. So how it's going to be occur? This is the part of initiation. What is there in this? This is the DNA strand. Along with that, this is the RNA polymerase, and along with that, this is the what? The sigma factor. When all of them comes, this is the DNA strand, this is the RNA polymerase, and this is the sigma factor. Now they have attached to this promoter site. Okay. Now they can move forward, and when they will reach a triplet codon, that is called as what? Initiation codon. At that point, this transcription will begins. Okay. So as the transcription will begin slowly, it will also elongate also. So that's why the next phase is what? Elongation. Once after this attachment of the RNA polymerase, whatever the sigma factor we have used earlier, it will get what? Removed also, right? So this is why you are seeing only RNA polymerase is attached at this initiation point of transcription, but not the sigma factor. Sigma factor will be subsequently released and it can just reused for the attachment of another RNA polymerase, right? So now let's see what happens with this RNA polymerase and this strand. So this is about the second phase, that is these elongations. The complementary RNA are synthesized in the 5' to 3' direction. That is direction anti-parallel to the DNA template. Okay, means what? Whatever this direction, whatever the direction of this RNA is there, that will be the 5' to 3' direction. Means what? The direction of this strand will be 3' to 5' direction. There are two strands, this one and this one. So the direction of this strand would be what? 5' to 3' direction. And the RNA which will be synthesized, that is in which direction? 5' to 3' direction. Okay. Let's see next. It does not require the primers, and no primers are means what is this is being said. We have seen in the case of DNA replication yesterday that we were required to for this DNA polymerase we required a primers or the primase enzyme complex, but here there is no primase or primers are required. No primase are required. Only not even primers or RNA primers are required to be formed. So that was the case with the help of in case of this. Okay. Now we will see the next factor, that is the DNA supercoiling problem solved by the supercoils solved by this topoisomerase. We have already seen what are these topoisomerase are there. So even if there is a supercoiling of DNA occurs in case of this or this any strand, that supercoiling yesterday we have already seen this topoisomerase. So in this case also that problem is going to be solved with the help of what? Topoisomerase. And what exactly this topoisomerase is doing? See, if this there is a coiling here like this, okay, then what this DNA topoisomerase will do? DNA topoisomerase will cut this strand here, and this entire strand can be now formed like this only, the previous or the untangled form. Okay, suppose this is the kind of the coiling which has been occurred. The DNA topoisomerase will come and it will cut strand somewhere here or here. After that cutting, this entire strand can be formed like this. So this is the exact function of the DNA topoisomerase. Now what? Let's see what happens in the termination part. See this termination part, you can see this elongation parts up to this end of the elongation. You can see this initial RNA polymerase what at this point. Okay, I will rub this entire annotation first. Okay, you can see this DNA polymerase and this RNA protein RNA synthesis start at this point. Okay, now it has moved further. Now it is about at where point? This point. Now it is about to come across this termination codon. okay, termination codons, which will just terminate this entire transcription. Okay, and you can see this initial RNA was like only this one. Now it has been elongated to a much longer length, right? So what is the next step that is going to occur? Now this whatever this entire transcription is there, now it's about to get terminated. Now this desired length or desired chain length of this RNA has been achieved. So now we are about to terminate this entire RNA synthesis. Next, the next thing is that to help that termination, there is another factor that is called what? ROHO, that is rho factor. Now this rho factor will attach with this entire complex, and as you can see, as this entire this transcription just uh, approaches this termination point, this rho factor will just terminate this protein here, this RNA, sorry, not protein. This terminate this RNA, so you can see these RNAs are here. Then it will also get separated from this entire complex so that they can subsequently use for the another termination of RNAs. Then this RNA polymerase is also going to be reused. You can see here, this is the RNA polymerase that was reused. So this what occurs at the termination point. Okay, you are getting these different RNAs. Even this mRNA, which was even this DNA strand which was earlier used, that is also, you can see it is also like here. Okay, so this is what happening in this transcription. You can see now just entire cycle can be just repeated. Okay, here again these are getting combined with this the RNA polymerase and this sigma factor. Then again this entire RNA synthesis is going to be occur. So this is the entire diagram of what? Termination. There is another case, that is this termination can also occur without the help of this rho factor. That is called as rho independent termination. And how this rho independent termination is occurring? That rho independent termination is occurring with the help of palindromic sequences in this DNA strand. And what are these palindromic sequences means? The palindrome means what? Suppose this is the name, that is a madam. Now you can use this, you can read this madam from this side, or you can read this madam from this either this side. In both directions, this madam or this term will sound same or will appear same. So such nucleotide sequences are also there inside the set, like CAC. Now this will be the same in the both direction. In this direction also it will be CAC, and in this direction it will also be the CAC. So such palindromic sequences of nucleotides are present at the end part of this DNA. Where they will present? They will be present in the case of where we don't have this rho factor for this termination. In that case, these termination points will have such palindromic sequences of nucleotides. And what will happen because of these palindromic sequences of the nucleotides? Whatever this RNA which is going to be formed is there, it is going to be form of hairpin-like structure. Okay, the nucleotide pairing with its own nucleotides. Okay, and because of such hairpin-like structure, this entire RNA synthesis is going to be terminated. Okay, so this is what happened in the transcription. Here is the detailed diagram of this entire prokaryotic DNA. You can just draw like here. This is the 35 sequence, Manstein Prinbo-Bosch sequence. This is the start of transcription. Okay, you can call this as an entire as a promoter sequence. Okay, so this will be the template strand. This will be the template strand, which will be read, and the RNA is going to be synthesized on this. Okay, so these are the all these prokaryotic transcription. Now let's learn about this eukaryotic transcription. So in this eukaryotic transcription, there are particular polymerases are involved. Okay, we don't know much about as such about them, but whatever the information we have, I am going to explain. Let's see the RNA polymerases. See, the RNA polymerase 1 is there, 2 is there, and the RNA polymerase 3 is there. What is this RNA polymerase 1 is doing? It is the synthesis of precursor for large ribosomal RNA. Okay, so the first polymerase will synthesize what? Large ribosomal RNA, large rRNA. What will be the second will do? It will synthesizes the precursors for mRNAs and small nuclear RNAs. Means what? It is synthesizing these mRNAs and snRNAs. Right. Now this third type. Third type of RNA polymerase will do what? It will participate in the formation of tRNAs and small rRNAs. Okay. So these are the different types of polymerase you will have to enlist in the case of this eukaryotic transcription. Now this is the diagram of this eukaryotic DNA. You can draw as such. Just the change will be this cat box will be there at its location is -70, and this Hogness box is there. Its location is -25 bases. If you remember these bases only, you can draw. Otherwise, just name the mention. Other than mention the names only. Okay. So let's see what are the promoter sites in case of this eukaryotes. These are the Hogness box, also called as Tata box, or the cat box. Okay. And both of these sites are helping what? RNA polymerases to binding. Okay. So this is the only limited part we know about it. That is the initiation part, that how it is initiated. Okay. And it is supposed that most of the other things, or most of the other elongation and termination part of these RNAs might be just similar like the prokaryote. So here I will only explain the initiation of this eukaryotic transcription. Let's see the transcription factors. There are different transcription factors which will bind to the DNA sequences in the promoter region. So this is that DNA strand. This is the initiation point, and these are the two promoter sites. So the transcription factors, all of the proteins will bind to these promoter sequences. Then there are certain other transcription enhancers are there also there, or the activators are there. Other proteins will get also attached, and this entire machinery will stimulate the transcription. It will get stimulate the entire speed of transcription. So there are 6 transcription factors are there which are doing this. These are called as this TF2A, B, D, E, F, and H. Okay. A, B, D, E, F, and H. The 6 transcription factors are there which are just activating or initiating this entire transcription. So this is what we know. So let's there are some other points which we know about this transcription in case of this eukaryotes. That is this, whatever this RNA is going to be synthesized after this entire termination, elongation and termination. Those RNAs in case of these eukaryotes are not like fully ready to be utilized. So there are certain things that are called as post-translation modifications that are required in the case of specifically eukaryotes. This prokaryotic RNAs which were earlier synthesized, okay, that were most of them are like ready to be used. You can just use them directly, okay, as such. Though no such post-translation modification is required. But in case of this, there is some requirement of the post-translation modification. So let's see one by one. So those RNAs which are synthesized in the case of eukaryotes, they are called as what? This heterogeneous nuclear RNAs, HNRNA. Okay, and these are the primary mRNA transcript produced by this RNA polymerase 2. Okay, so these are the primary RNAs which are synthesized after this eukaryotic transcription. They are called as what? HNRNA. Okay. Now they are required to process further. So what are these processes are going to happen? The functional mRNA is produced. This is the kind of inactive RNA. So its conversion to the functional RNA is going to occur with the help of what? The processes like this. 5' capping means what? Suppose this is the 5' end of this RNA. This is the 3' end. Then this 7-methyl guanosine group is added to this 5' end. That is called as what? 5' capping. What's the next stage of modification is that poly A tail. What is means by poly A tails? I mean whatever this region of tail region of this RNA is there, they at that region many adenine nucleotides are there. So it is overcrowded by this adenine nucleotides. So that is why it is called as what? Poly A tail. Then there is the next modification, that is the introns and their removal. How these introns are going to be removed? See, we have seen this part of this intron and exon are there, and that means that these introns are not having any specific meaning or any significance. So that's why these introns needs to be removed from this entire RNA sequence. What we want is that this only exon and see like exon 1 and exon 2. There can be another exons 3, 4 like, but only the thing is that we don't want this intron to be present in between. or even in RNA. So for that intron removal, there are certain proteins that are called as what? snRNPs are there. What these snRNPs are doing, they utilize the energy in the form of ATP, and they will just form attachment with these introns, and they will just remove this entire introns from this RNA structure, and they will bind this exon 1 and 2. Okay. Now you have got this entire intron separately and this exon and exon 2 separately. So what we have just done, we have just removed this entire introns from the RNA structure. Now this entire eukaryotic RNA is now what? Is now able to function correctly. So that's why this is one of the post post translational modification in case of eukaryotes. So this was the case with the case of prokaryotic transcription and the eukaryotic transcription. Now we are going to see there are not only mRNAs are synthesized, there are certain other RNAs are also synthesized. These are called as the transfer RNAs. Okay. In this transcription, and this how they are synthesized, just see, this is the structure of this tRNA. Okay. What is done with this? Just the 3' terminal of this tRNA or having this CCA end. Okay, suppose this is the 3' end, then at this 3' end, this CCA nucleotides are added. And what is the case with the ribosomal RNAs? In the case of this ribosomal RNA, first the pre-ribosomal RNAs are synthesized, and these are then later converted into what? Ribosomal RNAs. Only limited or the specific information we have regarding these tRNAs and RNAs, and whatever the information we have till now, I have just shared with you. Okay, this is the at the 3' end of this tRNA. After its synthesis, this CCA nucleotides are added, and in case of the ribosomal RNA, only the pre-ribosomal RNAs are synthesized first. Then these are converted into the ribosomal RNAs. There are certain inhibitors are also there of this entire transcription. These are the various drugs like this actinomycin D, antibiotics you can say, the pampine and alpha amanitin. So these are the antibiotics which will inhibit this entire transcription. Okay. And there is another concept like this reverse transcriptase. What we have seen is that the DNA, from this DNA RNA was going formed. But in case of this reverse transcription, what is happening is that a complementary DNA is synthesized from this RNA. So that's why it is called as what? A reverse transcription. And the enzyme that is responsible for this reverse transcription is what? This reverse transcriptase. Okay. So this is also called as what? RNA dependent DNA polymerase. So that is what happened in this reverse transcription. Okay, so this was what we have about this prokaryotic and eukaryotic transcription. This more detailed, yes, good evening Samina. More detailed notes obviously are there for the paid batch students. If you want the more detailed notes printed also and the lectures also, you can join that batches. Even if you are not attend, at least for the cell biology, you won't be able to attend the live lectures. There are only 2 lectures are remaining, so you can at least have attend these recorded lectures also. So yes, if you have any query now, you can ask. Do you have any queries? Otherwise, we will stop our session here, and we will meet on Monday 5 p.m. for our next session that is very important on the translation. Do you have any queries? I will wait 1 or 2 minutes because there is a delay is there always in between these online interface. So I will wait for 1 or 2 minutes. If Shravani is online, then I can answer her question also. If she is online, just ask me one more time. Any queries are there or no? If no queries, रितिका अजमेर सर प्लीज पीडीएफ हिंदी में भी प्रोवाइड करवा दीजिए. Yes, Ritika, I saw your comments after every lecture, but the thing is that I cannot I am not उतना भी एक्सपर्ट नहीं हूँ मैं हिंदी में, है ना. तो और ऐसे भी बहुत कम टाइम मिलता है, ये तो इतने ये लेक्चर थे ये भी नहीं ले पा रहा था. I was not able to even take these lectures all these days because of the paid batch schedule. But I have decided to at least have this half an hour session for this free lecture series in the evening. So we will have this continue, but I don't think it will be possible for me to provide the PDF in Hindi at all. I can use a part of Hindi in this lecture, है ना, कुछ पॉइंट्स जो है मैं हिंदी में पढ़ा सकता हूँ here, बट हिंदी में PDF तो पॉसिबल ही नहीं है ना, क्योंकि ऐसे ही मुझे कम टाइम है पेड बैच से। फिर भी मैं ऐसा लग रहा था मुझे कि हमारा जो रेगुलर यूट्यूब बैच है वो बहुत पीछे हो रहा है, इसलिए मैंने ये 5 to 5:30 का सेशन होगा हर रोज, only except these Sundays. At that we will have a session, short sessions on all of these points on the syllabus. Okay. So okay, we will stop here. As I can see, there are no queries. So we will meet on Monday, as tomorrow is Sunday. So we will meet on Monday 5 p.m. with our next chapter, that is the translation. Okay. So till then, have a great day. Bye bye. Good evening.